This is lecture 8, which is all about controlling microbial populations and, for the most part, making them smaller, so killing or inactivating microorganisms. There are two parts to this lecture. Slide set 1 looks at physical and chemical methods for basically controlling microbial populations on surfaces, in liquids, in foods, but not in people, not in animals. So controlling microbial populations in people is the second part. Slide set 2, antibiotics. So we'll get to those in a bit. But first, we will talk through a lot of the general concepts regarding microbial control. There's a lot to think about, and to be honest, for me, the most important thing I want you guys to learn is these concepts surrounding microbial control. There's some things in here that will probably change the way you think about techniques and products you use every day. So let's get into it. The first thing I want to mention is infective dose, and you might have seen this before. This is the idea that an infectious disease, which is caused by a microorganism, it will only happen if a certain number of those microorganisms get into you. So you have to be exposed to a certain minimum number of any microorganism before it can cause a disease. And in some cases, that number could be huge and really easy to avoid. So some of the weaker strains of Vibrio cholerae that cause cholera, you would be safe unless you consumed, let me think, that is 10 billion cells. And it's easy to avoid that if you have clean drinking water or any ability to treat your drinking water. And so the only time cholera appears is when people don't have the ability to treat their drinking water, and then cholera becomes a big, deadly problem. But in most situations, we are able to avoid eating that number of cells. And we can contrast that to lots of other really nasty pathogens that are more hazardous to people who live in places that have good infrastructure, like Shigella species or different E. coli species that can contaminate food. And 100 bacterial cells are a lot harder to avoid. One little spot on some spinach could have 100 bacterial cells, or one little area on a counter that didn't get disinfected could have 100 cells very easily. So it's hard to avoid a small number of cells. So if this is the infective dose for Shigella, it makes it much easier to catch a Shigella infection. So the reason I bring this up is that this is what we can think about when we talk about controlling microbial populations. If we're controlling the amount of microorganisms on a surface or in water, in the back of our minds should be, what infective dose are we worried about? Are we trying to protect against Shigella, or are we protecting against a weak cholera species? And that's going to be based on what do we think will be present and what historically has been present. So you'll see as we get to it. So there are a lot of other terms and concepts. These are important to know. In this video, I'm only going to talk about the ones that I will actually be using. The most important one is sterilization. This is the process of killing everything, destroying all forms of microorganisms. So if something is sterile, it doesn't have any virions, or none that are capable of causing disease. It doesn't have any bacteria. It doesn't have any fungi. It could have dead bacterial cells, it could have dead fungal cells, it could have inactivated virions, but nothing that can actually cause a disease or grow. And so some disinfection processes, some of the things we use aim for sterilization. You could imagine a needle and syringe used in a clinic has to start out completely sterile. And so in order to, so sterilization is required for things like that. And sterile is the adjective we use here. Sterilant would be something that achieves sterilization. And then we contrast that all with disinfection and disinfectants, because they destroy most organisms. Disinfection is the general process of destroying microorganisms and reducing their populations, but it's not sterilization. Disinfection typically refers to things that will kill vegetative organisms, so bacteria, virions, but not endospores and not the toughest viruses. Yeah. So this is a concept map from the textbook, and it's too small for you to see in the slides. It's not that important. But what it's showing is physical methods for microbial control, chemical agents, and then mechanical ways of removing microorganisms. And these black bars tell you which ones are capable of sterilization. That one should have a black bar. So, whoops. So the point here is they're showing how important it is to know whether a technique can fully sterilize something or not. Let's see. Some other techniques you would want to know would be like bactericidal versus bacteriostatic. So something that's bactericidal can kill bacteria. Something microbicidal can kill microorganisms. Something that's bacteriostatic stops them from growing, but it's reversible. So if you treat microorganisms with a bacteriostatic chemical and then somehow remove it or wash it away, they will start growing again. And so microbicidal is just a more general term, and microbistasis is a general term for not specifically bacteria. You can also have fungicides or, yeah, fungicides. Anyway, what else? Let's see. Decontamination is going to come up when we look at different chemicals we use. Decontamination would be physically removing microorganisms, like mechanically, like brushing them away, wiping them away, washing them down a drain, as opposed to something like sterilization that is destroying them, or removing all of them. Let's see, what else? There are several different techniques we refer to as asepsis, and that's when we remove pathogens. So we're not killing every microorganism, but we're removing all the ones that we think could cause a problem to a human. And so this would be like surgical, surgical asepsis would be preventing pathogens from getting to a wound. You don't have to kill every pathogen in the room, you have to make sure they can't get to the wound. And then aseptic technique is a term you would hear in the research laboratory. This is the set of techniques we use in the laboratory to handle microorganisms without introducing contamination and without contaminating ourselves. So it's how you control the location of microorganisms. That's what we call it in the laboratory, and if you're curious, I can connect you to the book chapter I wrote about aseptic technique 3 years ago. In medical settings, aseptic technique means a different thing. I mean, generally, it means limiting the spread of microorganisms and controlling where they are, but it's in a medical setting, you're not as likely to take a population of bacteria and move it from one place to another. You're more likely to just try to kill them or discard them. Okay, that's enough for this first video. We'll come back and look at modes of action in the next one.